Hey guys, welcome to my presentation. This is Noonan syndrome with multiple lentigines, also known as leopard syndrome type 1. So I'm very excited to present this to you guys. This is a genetic disease that I've studied a lot in the past. I've done some research on it, and I'm excited. I have a lot of technical information that I think is pretty cool to share with you guys. I'm going to try and bring it down to that sort of late high school, early college level, but you might have to bear with me at a couple points. I'll do my best. I'm also going to talk a little bit about how this disease has been modeled in mice and how you would sort of approach designing a therapeutic for this kind of disease and modeling a genetic disease in general. So let's get right into it. Also, I'm going to kill my webcam at various points during the presentation, pop in and out because my face is going to be blocking some of the information. So you might see me come in and out. But anyway, let's get right to it. Okay, so just a brief overview of the disease. So NSML is a rare multisystem disease caused by the mutation of the PTPN11 gene. I'll go into what that gene is later. But this disease used to be called leopard syndrome. It was a mnemonic device used to identify sort of common clinical conditions associated with the disease. We'll run through those really quickly. The L, lentigines, so those are sort of in the top right picture, those small brown macules that cover the body. As you can see, it's not just the skin, it actually can occur in the mucous membranes and even internally as well. The E stands for electrocardiographic conduction abnormalities. So if you see on the picture on the right, the most common heart abnormality you'll see is a bundle branch block on an EKG. Moving on, you have ocular hypertelorism. So that's in your bottom picture, so the wide set eyes. You can also see other facial abnormalities as well. P, pulmonary stenosis. So there are a lot of heart conditions that you'll see with this disease. Another one we'll cover is hypertrophic cardiomyopathy, but we'll get there when we get there. The A stands for abnormal genitalia. So in your male patients, you'll often see undescended or single testicles, and in female patients, you can see a failure to develop ovaries. The R stands for retarded or slowed growth, and you can see that very easily within the first year of life. And finally, D is for deafness. So moving on to sort of the more specific aspects of this disease. So the pathophysiology stems from the mutation of, once again, the PTPN11 gene. So what is that gene? It encodes for tyrosine protein phosphatase non-receptor type 11. Specifically, it's a protein called SHP2 protein, which is a protein tyrosine phosphatase. So what is a protein tyrosine phosphatase? It's a group of enzymes, and they remove phosphate groups from phosphorylated tyrosine residues on a lot of proteins. A phosphorylated tyrosine is a very common post-translational modification that affects protein stability, cellular localization, and it regulates the activity of various other enzymes. So specifically, SHP2 is ubiquitously expressed, and it regulates the RAS-MAP kinase signaling pathway. If you're unfamiliar, RAS and MAP kinase is a very important signaling pathway in the body. It is vital for cellular growth, development, and it is implicated in a lot of cancers. Any sort of dysfunction of RAS-MAP kinase is very bad, as you'll see in the previous slide, and also just in cancers, RAS-MAP kinase is always kind of mutated. So in NSML, the mutation affects the catalytic core of SHP2, the protein tyrosine phosphatase domain. So with this mutated version that you see in leopard syndrome, the catalytic core doesn't work. The enzyme does not achieve its basic function, and this greatly interferes with growth factor signaling. Looking where our gene in question is, so the PTPN11 gene is found on chromosome number 12. You can see that down here, highlighted in the red box, and it is specifically located at the location of 12q24.13, so this tiny red box over here. And it also contains 16 exons. Also important about our disease, it is autosomal dominant. You can see on sort of this pedigree here, so you have a 50% chance of passing it on to your offspring. So it is fully penetrative, so once again that's at 50%, but it has variable expressivity. So sort of depending on what residues, how much SHP2 you have expressed in the body, that kind of determines what sort of phenotypes you'll see in the disease. So moving on, I'm going to talk a little bit about how you might approach modeling this disease. So first of all, we're going to look at humans. So this is the human version of the gene. This image is pulled from the government database BLAST, which you can look up to see sequencing information, protein sequences, nucleotide sequences. So this is the actual nucleotide DNA sequence of our gene. So looking at it, we've got 16 exons and 15 introns present. Humans actually have 5 different isoforms of PTPN11, so you can have 5 unique transcripts, but the one that we're looking at right now is isoform 1, and that has a length of 91,000 essentially nucleotides. So if you were going to want to model this disease in a model organism, you know, to try and manipulate things and see what you can change, what's different, you'd want to compare the nucleotide sequence first. So quickly, I've done this with a couple of common model organisms. So mice in particular is very similar. So this is the mice version of our PTPN11 gene. So mice have the same number of exons and introns. They have 2 isoforms compared to humans 1, and their ortholog of this gene is located on their chromosome number 5. Going quickly through the other ones, so Danio rerio, that's the zebrafish, another very common model organism. As you can see, once again, the same number of exons and introns. Zebrafish only have the 1 isoform, and theirs is located on chromosome 10. You can also see they have a much shorter version of the gene. Moving on, these are 2 other common model organisms, but here we see a little variation. So C. elegans, nematodes, very common model organism for looking at growth and development. As you can see, they have only 6 exons, 5 introns, and theirs is on chromosome 2, so a lot of variation here. Very short gene as well, only 3,000 nucleotides. And finally, Drosophila melanogaster, that's the fruit fly. So they actually has an entirely different gene. They don't have a PTPN11, they have corkscrew, different name, but it's the same thing. It's a protein tyrosine phosphatase, and theirs is located actually on their X chromosome. So throwing this information all on the table, if you're looking at trying to maybe model our disease in a model organism, you want to see what's similar, what's different. So looking at this analysis, it would appear that the mice and the zebrafish would kind of be the safer bet here. So moving on, I'm going to show you something a little cool in my opinion. So this is, I actually wanted to show you the protein product itself. So this is a software called PyMol. This is what I use in my lab to look at protein structures. So this is a crystal structure of SHP2, our protein in question, and this is the wild type. So this is what you would have normally. So just kind of to orient you on what we're looking at here, as you can see at the top, you have in orange, this is the catalytic domain. This is what is actually mutated. And the important bit that we're looking at. The other bits are not as important for our discussion. So, looking here at this blue residue, this is what I have colored. This is the residue that gets mutated specifically. So right now, we have a tyrosine here. So, as you can see, I just want you to take a look at kind of what our protein looks like. Our enzyme, it is very open. This is the open conformation of the enzyme. So it is ready to take something in and cleave off that phosphate. So I'm now going to show you the mutated version, and then we'll do a little side-by-side comparison and see what you guys can see. But here is our mutant version. So as you can see, the blue residue has been changed into a cysteine. So this is the most common mutation that results in Noonan syndrome. And as you can see, so in our red is the catalytic domain. You'll see it a little bit better in the side-by-side comparison we're going to do. But this variant of the enzyme is much more condensed. This is the closed conformation. So this enzyme is catalytically, this enzyme is catalytically impaired. So it'll be actually unable to perform that phosphatase activity. And then very briefly, hang on, I'm going to do a side-by-side comparison so you can see that for yourself and not just take my word for it. Sorry, just resizing some windows here. Okay, so looking at the back, this is our wild type. You can see these, so these little corkscrew-looking things. These are alpha helices. These like arrows or beta barrels. You can see that the enzyme is very open here. There's a lot of areas for proteins to come in and be cleaved. And if you look at our mutant version, you can see how much more condensed and close together everything is, and it is unable to perform its basic function. But yeah, so this is kind of one of the cool bits that I wanted to show you. So moving on, I did a protein sequence analysis next of all of our model organisms, and just to quickly get that in front of you, instead of going through each one individually, we can see that looking at the protein sequence, so the amino acid sequence, not the gene, that the mice has huge similarity between humans, the human variant and the mouse variant of the protein. So given that information, it would make the most sense to look at mice as a form of model organism to study this disease. So true to that information, there have been a lot of studies of this disease with mice, specifically looking at hypertrophic cardiomyopathy. So that's kind of the most common heart condition associated with the disease, and is the most dangerous. It's a thickening of the heart wall, and it prevents normal conduction of blood. So most studies have used mice as their form of model organism to create, and they've created a knock-in model, making the mice actually have the mutation. And we're going to look at how they sort of looked at therapeutics for that. But looking on our right, we can see that the mice, here's the wild type on the left, here's the one with the leopard syndrome, and as you can see already, these mice have distinct face and nose profiles. How they made the mice very quickly, this is something very cool in molecular genetics. This is called Cre and LOXP recombination. This is how they made the mice. So this is a very versatile site-specific recombinase technology, and you can use it to make your own deletions, insertions, translocations, and inversions at specific DNA sites. LOXP are these specific sequences you would insert with another editing system, such as CRISPR-Cas9. But so you put in these LOXP sequences, and then you introduce Cre, which is an enzyme. And depending on where you put the LOXP, the Cre will either cause a deletion, an inversion, or a translocation. In our case with the mice, they did a translocation to introduce that sort of mutant sequence and cause their mice to have leopard syndrome. So moving on, these are sort of the phenotypes that they were able to see in their mutant mice with leopard syndrome. So as you can see, you have this thickened heart wall between the normal wild type and the leopard syndrome mice. You can see much thicker muscle walls there in sort of all of these. And you can also see delayed growth on our right here, and you can see sort of the stereotypical chest or skeletal abnormalities. And then sort of looking at future direction, modeling sort of a genetic disease in a model organism. So in their study, they were actually able to test rapamycin, which is an AKT inhibitor, which you can see here. Rapamycin is a mTOR inhibitor, and they were also able to test an AKT inhibitor, which is in this sort of RAS-MAP kinase pathway that I talked about. And they were able to actually see a reversal of the heart condition, and that was wonderful. If you were going to perhaps research this a little further, also looking in the pathway, BRAF and MEK are also parts of the RAS-MAP kinase pathway that are commonly inhibited in cancer and other diseases. So you might want to test inhibitors to those to see if that can reduce some of the phenotypes seen in NSML. And you can also perhaps elucidate more into the signaling pathways. And I would recommend that future studies into this use the mouse model, because as we saw, they had a lot of homology both in the nucleotide sequence as well as the protein sequence. And yeah, that's it. Sorry if that was a little fast. Had a lot of material I wanted to go through, and hopefully it all made sense. Let's see, webcam. thank you guys for watching, and feel free to post any questions in sort of like the discussion comments below. Thanks, guys. Appreciate it.